Sorry, my voice is a little bit coarse today, so I hope you can all hear me when I'm talking. Hi, my name is Hannah Hiraki, and over the course of this year in independent research, I've been researching rat lungworm, otherwise known as Angiostrongylus cantonensis, in snails on specifically Oahu's windward coast. Before I get started, I'd like to give a big thanks to Dr. Debbie Milliken, who's right here. She's my mentor, and she actually introduced me to this project and got me really interested in rat lungworm, got me the connections that I needed at the Bishop Museum and guided me through this whole thing. And also Dr. Yvonne Chan, who is my research class advisor. She's helped me through every single hurdle that I've encountered, and there were quite a few, and I'm really appreciative of both of them. Okay, so what is a rat lungworm? So, rat lungworm is a parasite that originated in Southwest Asia, and it tends to congregate around warm, wet areas, so tropical regions around the equator. It has spread to various parts of Central America and the United States, and has been especially rampant in the Hawaiian Islands, as you may have heard of different cases of rat lungworm disease popping up, especially in the Big Island. Rat lungworm is dangerous because it causes this infection called eosinophilic meningitis in humans through the ingestion of snails that contain these parasites. And symptoms of this infection can include nausea, nerve damage, and at worst, death. And as of 2008, 2,000 people have been infected in 30 countries worldwide. And to relate this back to Hawaii, in 2017, 18 cases alone of rat lungworm disease were reported in Hawaii, along with 2 cases in this year alone. So in the past 4 months, there's already been 2 cases. So the life cycle of a rat lungworm parasite depends on a series of different hosts in order to infect a human as a vital product, I guess. So the primary host is a rat, and it ingests a rat lungworm larvae, which matures in the brain and the arteries, and then it lays eggs that mature into larvae that are swallowed by the rat, pass through its feces. And this infected feces is then eaten by snails, which is the intermediate host. And when the larvae in the snails matures to the 3rd stage of its life cycle, of the larvae life cycle, if they're eaten either raw or undercooked, like hiding in your lettuce somewhere, if they're eaten by humans, then the larvae travel to our central nervous system and they cause an infection, also known as eosinophilic meningitis. So in terms of Hawaii, eosinophilic meningitis has been reported on 5 of the 6 main Hawaiian islands: Oahu, Maui, Kauai, Lanai, and the Big Island. And a 2018 mapping of the prevalence of rat lungworm in different snails that were collected along the 6 Hawaiian islands found that there were infected snails on Oahu's windward coast only, along with various locations on the Big Island, Maui, Molokai, and Kauai, but none on Lanai. But you can see, like, on those different maps, the dark spots represent the places that they found an infected snail. So a specific 2014 study by researchers at the Bishop Museum surveyed snails from 6 Hawaiian islands, and they separated each of them by snail species. And they found that overall, 6% of snails in Hawaii had rat lungworm parasites with a sample size of around 1,271. It's a pretty extensive study. For Oahu specifically, we only had around a 10% rate of infected snails, but it's mainly lower than other islands such as the Big Island or Kauai that had 33 or 34% infected snails respectively, but higher than ones like Lanai, which had 0%. Specific species were found to have higher and lower infection rates, with the highest being the semi-slug known as the Parmarian martensi, which is actually been found to be a very effective vector for this parasite, and it's probably one of the main factors contributing to such high levels of rat lungworm and rat lungworm disease in Hawaii. And so essentially the species of a snail could be used to determine how high the risk is of that snail having this parasite in them. So I asked the question, how does snail species distribution correlate with the presence of rat lungworm in snails from Oahu's windward school gardens? And I focused my research around the windward coast because I knew that would give me the highest probability of actually detecting rat lungworm in Oahu. And also I wanted to target school gardens because school-age children have a high risk of probably eating one of those infected snails, and I really wanted to determine like the risk at different schools and try to reduce that. And I also wanted to ask, how does it compare to past studies of rat lungworm in Hawaii? So the purpose of my research was to map out the snail species distribution in the Windward Garden schools, and I also wanted to detect the presence of rat lungworm, or the lack of presence of rat lungworm in all those snails in each of the school gardens tested, and I wanted to see the difference in the different species as well. So I hypothesized that the prevalence of rat lungworm in each species collected from the Windward School Gardens would correlate with the results from that prior 2014 study that I was looking at. Okay, so to begin, I collected snails from Iolani's Lower School Garden and two Windward School Garden sites with around 50 snails at each site. Iolani's school served as a control, and I was actually able to request help from the same researchers that were working at the Bishop Museum on this 2014 and 2018 studies, so that I would be able to have consistent methodology, and so that would help me to more accurately compare the results of both studies once I had my results. Snails were collected for 1 man-hour, basically 1 hour per qualified researcher there. So if there were 2 qualified researchers, we'd collect for 30 minutes. If there were 3, then we collect for 20 minutes, and so on and so forth. And this was done to collect an accurate snail species distribution at every single school to remain consistent, so I could compare them later on. And after collection, snails were heat shocked, so they were placed in boiling water and instantly killed, and then preserved in 95% ethanol. Back at the lab, snail tissue was extracted from the snail foot, which actually has the highest concentration of parasites on an infected snail's body. The tissue was then ground and placed in a lysis buffer, and it was incubated to break it down. And I was able to extract the DNA by isolating DNA through different washes of buffers and other solutions that kind of got rid of the fats and the sugars and the proteins along that was within the sample. And I also included a positive snail, which served as a positive control from the Bishop Museum, so that I could later track to see if my samples actually compared to the positive control that contained those rat lungworm parasites. Okay, so next I added species-specific primers that were specific to this particular ITS1 region of the parasite DNA. And I also added other ingredients into a thermocycler for PCR that uses temperature cycles to amplify DNA using DNA replication, basically giving it all the ingredients to replicate itself. And I was also running this with the positive controls that I extracted earlier. The samples that were PCR'd were then run through gel electrophoresis, which is a method that runs the samples through an agarose gel using electrical currents, and these currents separate DNA by different sizes, so I could identify the fragments of DNA, and the dye would then bind to those pieces of DNA that were specific to the rat lungworm DNA in order to detect it. If you can see on the bottom right picture, the black bar, the black band that has the plus sign above it is a positive control. You can see how it really shows up on the gel, and that shows the presence of this rat lungworm DNA, and that differentiates it with just snail DNA that would have been found in the sample anyway. And I have full confidence in my results because I ran all these reactions with positive controls to ensure that my PCR had worked and it had actually amplified that DNA. Out of the 147 snails that I tested for rat lungworm, I found that 0% contained the parasite. And just for reference, the highlighted region right next to the bar graph is the percentage of snails that were found to have rat lungworm per species. So yes, all of them right now have 0%. So I compared the results of this experiment with the past 2014 mapping from the researchers at the Bishop Museum, and I found that the infection rates of the bottom 3, Bradybina similaris, Veronicella chimensis, and Sabulina octana, were relatively low to begin with, even with the results of their study. But however, the top one, Lisacatina folica, was quite high with an infection rate of 10%. compared to mine of 0%. And I'd like to mention, though, that the species distribution of both were different because they were different studies, because I had different amounts of snails per species in each study. It was hard to accurately compare and proportionally compare the two studies. So I used a Fisher's exact test to measure the statistical difference between the results of my study and the 2014 study. And I found that the infection rates of the Bradybina similaris, Veronicella cubensis, and Subulina octona were not significantly different than the results from the 2014 study. However, the prevalence of rat lungworm in the top one, Lasacatina folica, in the 2014 study was significantly different than my results. Some explanation behind this could be the fact that their study in 2014 evaluated all 6 of the main Hawaiian islands. So this prevalence level, that 11% prevalence level that they found, was representative of all the islands, whereas mine was only representative of Oahu's, and Oahu generally had lower detection rates compared to islands like the Big Island or Kauai, and that could have probably brought my infection rate down compared to the ones on the Big Island. Yeah. So in conclusion, no rat lungworm was detected from the 3 school garden sites that I surveyed. And in general, the snail species that were found in each school had historically pretty low infection rates in Hawaii in the past studies. And the presence of rat lungworm per species were not significantly different for 3 of the 4 species in the 2014 study. Because each species had an infection rate of 0%, I was unable to compare the species within the study as I had hoped to do before. And now significance. So by conducting this research, I determined the threat of rat lungworm in a windward school garden. And I think in the future that can definitely help to gain insight into developing different safety measures that can help to aid high-risk schools. As you can see, these are very low-risk schools, but if there were to be one with a lot of infected snails, we could take those precautionary measures that would be determined by this research. Also, the lab procedures and the field safety measures that were used in this experiment can be repeated around schools on Oahu's coast and other school gardens, which would help to expand the sample size, and it would definitely bring more accurate results for this research and better map out not only the rat lungworm prevalence around Oahu, but also map out this the snail species distribution as well, which there's not that much data on in Hawaii. And this study in general also spreads awareness of the risks of consuming raw snails and developing rat lungworm disease. And if tested schools can take different extermination measures against snails, then they definitely will lower the risk of school-age children being susceptible to having one of these fatal infections. And it could prevent further deaths or injuries in the future. Oh, and also, in terms of extermination, we don't have to worry about native snails dying because basically most of the infected snails are invasive, so we don't have to feel too bad about exterminating those. That's a little tidbit I had to add. Okay, so for future research, I definitely love to recollect and test my current sites to confirm those results, and as well as more accurately map out the snail species distribution and rat lungworm prevalence of the different schools. I also hope to expand my sample size. Of course, as I mentioned earlier, it would help to better map out a larger area of Oahu rather than just 2 or 3 schools that I had done in this research. And it would also decrease the amount of error. And finally, I'd hope to observe different snail and rat extermination protocols and try to see if the elimination of the hosts of this rat lungworm would decrease the prevalence of it around Oahu. So what did I learn from doing research this whole year? The first and main thing that I learned was perseverance. So early in my research, I worked with a faulty positive control, and I basically had to test every single one of my samples and rerun them and re-extract them a second time, which was a huge time commitment and definitely a little tough on me. But I learned to persevere and just be determined and get to the end goal and finish and feel that accomplishment at the end. I was also able to refine my lab techniques and troubleshoot because I spent so much time in the lab. I picked up on efficient lab techniques that will definitely prove to be very useful to me in the future if I plan to pursue research as a career or maybe just in college. And I learned to troubleshoot because I did run into many different problems. And I also learned the importance of positive and negative controls. Yes. And finally, don't be discouraged by results. I was selfishly hoping to detect at least one snail with rat lungworm. I didn't. But in general, it's a good thing. And I know I had valid results, and I'm just going to hope for the best next time. But I've learned to accept the results and, yeah, don't doubt my results. Special thanks to Ken Hayes, Noreen Young, and Janie Kim, who are researchers at the Bishop Museum that had conducted those past studies and helped me in terms of methodology, troubleshooting, all sorts of things, resources. I'd also like to thank the Bishop Museum for lots of resources, information, and guidance. Dr. Debbie Milliken, again, my mentor, and Dr. Yvonne Chan for just pushing me through this whole thing. Mr. Eric Tong, who works in the wet lab, and he actually helped to allow me to get some more lab hours in during winter break or on the weekends, just so I could finish running all my samples again. And finally, my research class, because we were all in the same boat. We were all pushing through that too. Okay, thank you.